prior to some technical difficulties, we had been talking about the lac operon, and I wanted to point out that an operon is just a group of functionally related genes. And so a part of this is the LAC-Z gene, and this codes for an enzyme called beta-galactosidase. And what it does is it's going to break the glycosidic bonds between glucose and galactose. It's going to, or I should say that it catalyzes the breakage of these glycosidic bonds between glucose and galactose. And this is, this is something that happens every time that bacteria are exposed to amounts of lactose. The gene gets turned on. But one thing that we can do is we can expose beta-galactosidase to something called XGAL. And XGAL is it's normally colorless and is a synthetic sugar, so it's something that's man-made. We made it. It doesn't exist in nature. But whenever we expose XGAL to beta-galactosidase, it turns blue. It results in a blue byproduct being produced. And so I just wanted to show you I guess a visual representation of this. So here we see the breaking of the code from the restriction endonuclease comes in, that we can insert our foreign DNA. These are our ampicillin resistant genes, and this is the LAC-Z gene. Because where we're going to insert the foreign DNA that is going to take the place of the LAC-Z gene. We're going to cut out that LAC-Z gene. We're going to insert this foreign DNA. And if, say, we're dealing with a plasmid that took up or a bacteria that took up the original plasmid, or the plasmid that these ends just kind of resealed back together, then obviously its LAC-Z gene is going to be functional. It's going to be able to produce beta-galactosidase. And so beta-galactosidase, whenever it's exposed to XGAL, is going to produce this blue byproduct. And so that's what kind of leads on to this. So what we do is, and I suppose I was getting ahead of myself, we take ourselves a little petri dish here, we expose it to a hot nutrient liquid called agar. We throw in some ampicillin to kill off the bacterium, and then XGAL. And we incubate it in a period for 24 hours. This is a very, nutrients, it's away from fungi. It's an optimal, circumstances for bacteria to undergo binary fission. And I want to remember that they have very short generation spans. Every 20 minutes or so, they undergo binary fission. So after 24 hours, we're going to have colonies of bacteria grown in this petri dish. And so what we have is some of these bacteria that are blue. And if they're blue, what does that have to say? That they're exposed to the XGAL, the synthetic sugar. And whenever that XGAL synthetic sugar meets up with beta-galactosidase, it results in this blue byproduct that we can see here. And the problem with this is that means that these have the original plasmids, or their plasmids that were broken up by restriction endonuclease were then resealed, and they have therefore no foreign DNA. Also, there's these green colonies here, and these are definitely the normal bacterial color. And it could be green, it could be white, it could be whatever. I drew it green because I always think of bacteria as green. And these are the ones that have taken up some type of gene, because we know that, one, they survive the ampicillin, because the ampicillin is that the ampicillin kills the virgin bacteria. Let me rewrite this. That is the ones with no plasmids. The bacterium that did not even take up a plasmid. And I always like to think of this as a slasher film. If you've ever seen, and I don't know why you would unless you're really stupid like I was when I was a kid, but there were really crappy slasher films about dudes running around in masks. Very brilliant drawing there, I know. Who would run around with masks, and he would always stab people, and it's, hey, let's go to the lake to make out where there's nobody there. But the first person to die always is the virgins. The virgins die first in every movie. Glasses. Ooh. Glasses, the virgins die first. And I guess you could think about the rituals or whatever. But anyways, ampicillin kills the naive virgin bacterium. So that's why we killed off all the naive virgin bacterium with ampicillin. We exposed the XGAL, meaning these are the ones that have taken up the plasmids that resealed and have no foreign DNA. And these green ones, these normal bacterial colors, are the ones that have either the right gene or the wrong gene. They took up at least some type of a gene. So blue took the original plasmid, the ones that have no foreign DNA, and then the original color, which is because they're alive and they're not blue, they're the ones that took up plasmids with foreign DNA. And then the question being, okay, of these normal colored bacterial cultures, which one of these has the gene of interest? Which one of these has the wrong gene? And that's what leads to a question, and it would originally be a pretty difficult one to answer, except for when we're looking for something, we have to make a nucleic acid probe. And a probe is just something that it's what we use to look for something. And so if you've ever you've ever been to the doctor for any type of lower colorectal type issues, what do? They make you get up on all fucking fours and shove this little probe thing up your ass so far that you're screaming and hollering in pain. It's, oh, look what's in there. No dignity for the sick. No dignity. Ugh. Anyways, my point being that a probe is just it looks for something. I suppose I could have drawn a space probe. You got your little whatever rocket ship thing, flies off, lands on Mars, and what does it do? It looks around for stuff. Particularly, it's looking for water, but that's not really important. It's Mars. It looks around, and that's what a probe does. It looks for something. And so, what are some features that we want to have about this probe? Well, it's got to be small. It can't be the entire long nucleotide sequence of this gene. It's got to be complementary with part of the gene of interest. So say your gene of interest has a part of it that's T, A, C, G, whatever. Well, then the probe has got to be C, G, T, A. It's got to be complementary with part of the gene of interest. And I think you should really put these two terms as kind of connected to each other, because if it's going to be complementary, meaning it's going to bind to that gene of interest, it's got to be single-stranded. It can't just be, if it's double-stranded, that's not going to do us a whole lot of good. And it has to be radioactive, because what do radioactive things do? Well, they glow. So if something is glowing, plutonium has been exposed to it or whatever, we can trace it. We can, see what's where. So how do we figure out, how do we make this nucleic acid probe? Well, one of the things that we're going to do is we're going to think of what our gene of interest is, and we're going to say, one of the gene that codes for a protein, testosterone or insulin or whatever, and then we're going to plug it into an automated DNA sequencer. And what the automated DNA sequencer does is it tells us the sequence, hence the name, on the gene of interest. It's going to tell us the nucleotide sequence that is on the gene of interest. So we plug it into the computer, it's going to blah, 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 it's going to tell us the nucleus, so we'll say that the nucleotide sequence is A, T, A, C, G, C, whatever. Keep going. So whenever we make our probe, we'll take this. We now that we have the nucleotide sequence, we're going to take this and we're going to plug it into the automated DNA synthesizer, which what does a synthesizer do? Kind of like the word synthesis means to make, to combine things. So it makes a probe, makes a model probe. Makes the model probe. And so what we can do is we take this small model probe that we've made from a DNA sequencer telling us what the thing was, and then the synthesizer, which is what we plug in here. This is it's supposed to be a factory. I know it's very hastily drawn, but we make it, and it's going to make one molecule of a model probe. And so what we can do is we can then expose that to the gene of interest, and since it's single-stranded, it's small, it's complementary, and it binds with it, it's going to bind. Now this, I want to make a point here, this is not radioactive yet. It isn't glowing the moment it comes out. But what we have done with the automated DNA synthesizer is we have made one molecule of the model probe. Okay, skipping over there. And what we can do with that, though, and I'll have to kind of resort back to this picture here. One of the things that we're going to do before I continue on is we're going to take pieces of these green or original or normal bacterial color colonies out of the petri dish, and we're going to put this into things called an ASI plate. And now everything is going to have these little wells, and so you can select B2 or B3 or whatever. And what we're going to do is we're going to take small amounts from original color colonies and put them in an ASI plate. And so we put these small amounts. We're going to expose these things to a chemical that's going to lyse these cells. And lyse, remember, means to rupture. So they just go, and it's going to all this fluid is going to leak out of it. And then once we do this inside of these wells, we're going to add an enzyme that's going to hydrolyze. What does hydrolyze mean? To break bonds by adding water. Why I drew a water molecule. It's going to hydrolyze these hydrogen bonds between the DNA. So we go from having this double-stranded DNA, this stable double-stranded DNA inside of the cells of these acyclate wells, and it's going to be single-stranded. Once it's single-stranded, though, we can add these copies of the probe that is going to bind with the gene of interest. So we put in a probe that's going to bind with the gene of interest, and voila, it's bound. Now, I want to make sure that we only have one model probe at this point, and it's not yet radioactive. So how do we get to where we can see, in mass quantities, where we can look at this and then it'll literally glow what points we're looking for? Well, that's what this thing called polymerase chain reaction comes in, and the guy who did it is a billionaire, and it's been trademarked. I should probably trademark. Whatever. And so the steps of polymerase chain reaction, the first thing we want to do is you want to denature the molecules of the bound with the probe. And I drew this, I don't know if you can tell, but that's supposed to be green, but let's draw it like this. So first thing we want to do, we want to take this now double-stranded DNA with the probe on it, we want to heat it up to 95 degrees Celsius. Once we heat it up to this high temperatures, it's going to denature the DNA. It's going to break those hydrogen bonds so that we have single strands. This is our gene of interest, this is our probe. This is our target sequence, I guess you could say, is another word for the probe here. So we heat it up 95 degrees Celsius, almost boiling temperatures, and they have now split the bonds between the hydrogen bonds holding the probe and the DNA sequence together. We can then cool it to 55 degrees Celsius, and here we're going to add some primers. And primers they're the unsung heroes of this reaction because they're excruciatingly important. So we add these primers here that are going to bind to both the original DNA sequence and the model probe, and then we're going to add Taq polymerase into the solution here. Taq polymerase, because 55 degrees Celsius is still very, very hot. I mean, if my body temperature would be that way, my brain would be fried. Human body temperature is 37 degrees Celsius. This is 55 degrees Celsius. This is a substantial amount. This is pretty hot. So what we do is we got to have something that can survive these excruciatingly high temperatures. And what we use is something called Taq polymerase. And what the Taq stands for is Thermus aquaticus. And what these are is these came from the Yellowstone National Park, where it's 55 degrees, or even I don't know how hot the little geyser things can get. But what we do is we can take